The structure of DNA is that it has a double helix, and this structure is very important for both its function and how it's going to be replicated. In terms of the original studies of this, some X-ray diffraction studies were performed by Rosalind Franklin, and she performed these on DNA, and what she found was a 3.4 angstrom periodicity. We'll talk more about that, the importance of that in just a minute, and the characteristics of a helical structure. So what it shows is that the structure would repeat itself every so often. Watson and Crick in 1953 used much of her work to form their own theories, and what they found was that, or what they proposed, is that DNA is a right-handed double helix, the two strands are antiparallel, and the bases are stacked on one another. The two strands of DNA connected by these A to T bonds and the G to C base pairing, and that there are 10 base pairs per helix turn. So let's look at this a little bit more closely. There's a couple of really important parts I want to point out. So first of all, one complete turn of the helix, you can see how it's kind of turning here, okay? One complete turn from start to finish, let's follow this backbone here, start till it comes back to that same point, is 34 angstroms. Angstroms is just a very small unit that we will use to measure these very, very small things. Diameter of this is very consistent, 20 angstroms. And it is the reason why a pyrimidine will bind a purine and vice versa, but a purine will never bind a purine and a pyrimidine will never bind a pyrimidine. And the reason for that is because the size differences. If you had a pyrimidine with a pyrimidine and you had a purine with a purine, the diameter of these two base pairings would not be consistent. And so to keep this consistency of structure and of size, you need to have a pyrimidine binding to a purine. You will also notice, if you look a little bit more closely, that we have what's known as a minor groove and a major groove. This double helix is not perfectly even. There is a smaller bend between them and then a larger gap between them. The major groove is 22 angstroms. The minor groove is 12 angstroms. Because there are 10 base pairs in one complete turn, the distance between one of the base pairs is 3.4 angstroms. And that's just the distance in this backbone between the sugar here and the phosphate to the next sugar. And like I mentioned before, one complete turn is 10 bases. You can count them out here. We'll start here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And we can measure this, and we know that it is 34 angstroms. We have marked here the 5' to 3' end. If you follow this, this is the one backbone. The other here, you could start here at 5' to 3'. The importance of this, again, just comes into the structure and the way that the bases and the way that the phosphate and everything and the sugar is oriented. So within this double helix structure, we know that A's bind to T's and G's bind to C's, because this provides complementarity of the two strands and a very good chemical stability to this helix. We know that between the A's and the T's there are 2 hydrogen bonds, and between G's and C's there are 3 hydrogen bonds. And like I mentioned before, A can't bind with G, C can't pair with T, because that would lead to a whole variety of diameters in the helix. The sizes of purines and pyrimidines are different, and this would lead to instability in our double helix. One of the major characteristics or hallmarks of DNA is that it is very, very stable. And the only way to get this is by pairing the two different types of nitrogenous bases. There are several different forms of DNA that have been found. However, I'm only going to briefly mention them because you may come across them in some of your other studies. As far as I am aware, they are not thought to be biologically relevant. However, you can create these by combining the molecules in a test tube. So you can see here the structure, the bend of these. There's a B DNA, an A DNA, and Z DNA. Here with the Z DNA, you can see the distance between the helices is quite large. The A DNA just kind of bent a little funny, and the B DNA is backwards here.